Hi, welcome back to part 2 of your notes. We're going to pick up where we left off talking about Mendel's laws of inheritance. If you remember, when we talked about that, was about 1860s when Mendel came forward with this idea, presented it. Actually, when he presented it, it was viewed pretty skeptically because there wasn't really this understanding yet of what DNA was. There was lots of speculation about inheritance, and people wanted to know and were studying it, but Mendel's laws of inheritance were viewed kind of skeptically and kind of put aside until later in the 1800s. 1875, the process of mitosis was worked out. In 1890s, the process of meiosis was worked out. And it wasn't until those processes were worked out that scientists started to look at what Mendel had proposed and said, wait, maybe he was onto something. And that's how we got the chromosomal theory of inheritance. So what is the chromosomal theory of inheritance? Well, that is that Mendelian genes have specific loci or locations along the chromosome, and it is the chromosomes themselves that undergo segregation and the independent assortment. Okay, so the chromosomal theory of inheritance states that the behavior of chromosomes during meiosis and fertilization account for the patterns of inheritance. Okay, so take a look at this picture. Just real quick, laws of segregation, or the law of segregation, is talking about the two alleles for each gene are going to be separated during gamete formation. So we're looking here, let's just look at the R's. R's are going to stand for round seed shape versus wrinkled. Okay. The R's are going to be separated so that when gametes are formed, they can have either a big R or a little r. Voila. That's law of segregation. Alright, law of independent assortment says that alleles of genes on non-homologous chromosomes, okay, so chromosomes that aren't their pair, their copy, like we have here a pair or homologous chromosomes, one from mom and one from dad. Okay, so we're talking about alleles that are on different pairs. Okay, that those are going to assort independently during gamete formation. That goes back to what we were talking about meiosis, talked about independent assortment, that you're not going to have this nice lineup of all of mom's chromosomes on one side and all of dad's on the others. Okay, we want to have lots of mixing so that we can increase genetic diversity. Okay, which if you take a look here, we have lots of possible genetic diversity. Okay, one of the really cool things is if you think about humans, okay, we have two pairs of 23 chromosomes, which means for humans, there's a total of 8.4 million possible formations of gametes, or combinations, there we go, of chromosomes. Okay, then if you want to be really cool, and when you take 8.4 million, times it by the other 8.4 million that comes from, let's see, one from egg, one from sperm, you have 70 trillion possible combinations of DNA. Oh, I don't know why I did that. Okay, 70 trillion possible combinations of DNA that make you or me. So you truly, truly are unique. You are 1 out of 70 trillion combinations of your mom and dad's DNA. Awesome! Okay, so now we need to move on. I'm sorry, I'm getting bogged down in stuff that's not important. Well, I mean, you being unique is important, but we need to focus on learning targets. So, number 1, explain how genotypes relate to phenotypes. Okay, so what is a genotype? Genotype is how we're going to spell or represent these alleles. If you go back to Mendel's laws of inheritance, we talked about dominance and recessivity. Capital letters mean dominance, or going to represent, I don't mean, but they're going to represent our dominant allele, and the lowercase letters are going to represent our recessive alleles. So you can have different combinations. You can have homozygous. These are a homozygous genotype that is going to be allelic pairs that are spelled the same, or in other words, you're going to have all capital or all lowercase, or you can have heterozygous, and those are going to be pairs that are quote-unquote spelled differently, or in other words, have a capital and a lowercase letter. Okay, now what's phenotype? Wait, hold on. Genotype is referring to the genes. Okay, the actual DNA, what's being coded for from the DNA. Phenotype is going to be physical characteristics. They're going to be, can refer to function, can refer to appearance, something physical. Okay, so how does genotype relate to phenotype? Well, genotype is causing the phenotype. The genotype is the gene that is coding for red flowers, or for purple flowers, or for brown hair, or for green eyes. Okay, the physical characteristics like those green eyes or the brown hair, those are phenotypes. Okay, so learning check. If black is dominant, what is Fido's genotype? Okay, so I suggest whenever you hit a genetics question where you're asked to determine genotype, you write off to the side somewhere you've got to have a key. So we're told that black is dominant. Dominant is a capital letter, so big B is going to be black. Little b is, let's just say, white. Okay? So what is Fido's genotype? Well, he's black, so we know we've got to have a capital B in there. Okay, now there's actually two possibilities, and without looking at offspring or parents, we actually aren't going to be able to figure out which one it is, so I'm going to write down both of them. He could be big B, little b, depending on what mom and dad were, or it could be big B, big B. Okay, so how do we figure that out? Well, we had to do something that's called tracking generations. When we track generations, we're going to start with the P1 generation. These are the parental or the pure strains. Okay, pure strains are always going to be homozygous. Okay, big capital, both capitals or both lowercase. So if you take a look over here, I've got two guinea pigs, one with rough fur, one with smooth fur. So we're going to do R's. By the way, I would always suggest staying away from the letters that is difficult to tell if it's capital or lowercase. So that would be like S is not a good one. P is not a good one. W is not a good one. There are other ones. Okay, just use something that's very obvious to tell if it's capital or lowercase letters. Okay, then there's F1 generations. This is the first filial generation, or it's the offspring of the P1 generation, okay? So these down here, if you take a look, what do you notice about the capital and lowercase letters? You should notice that each guinea pig has a capital and a lowercase. They get one capital from mom, one lowercase from dad, or vice versa. Okay, then how we really track generations is we take a look at F1 generation. They should all be heterozygous. So we take two heterozygous, we breed them, cross them together, and we get what's called the F2 generation. F2 generation is where you're going to see all of the possible genotypes. So you're going to have homozygous dominant, you're going to have heterozygous, and homozygous recessive. Okay? So, next learning target, we get to talk about Punnett squares. Okay, 4, predict the probability of the occurrence of a specific trait by using monohybrid crosses, and 6, predict the probability of the occurrence of specific traits in offspring by using dihybrid crosses. Pretty sure you've done monohybrid, not sure if you've done dihybrid. We'll definitely go over these more in class. This is just kind of a brief introduction. Okay, so what are the types of monohybrid? This is looking at just one. Mono meaning one. Okay, one set of alleles or traits. So the picture to the left, we're looking at one trait, and that's color of the flower. Okay, then we have dihybrid crosses, which is we're going to look at two. Di meaning two. Okay, so two sets of alleles at the same time. Dihybrids are a little bit more complicated, but they're not that difficult. Okay, so where did Punnett squares come from? Well, they were developed by this guy named Reginald Punnett to help students predict offspring. Okay, there's a couple rules that you should follow when you're setting up Punnett squares. Maybe you knew them, maybe you forgot them, maybe you never learned them, I don't know, but here they are for you, okay? Females should go on the top of the square. So here, those are eggs that are on the top because the females have eggs. Males go on the side. Male, the sperm, so sperm on the side there. Okay, capital letters should always come first. Okay, and then we have what's called a test cross. You'll be asked a lot in practice questions to do what is called a test cross. Test cross, you take an unknown genotype and you cross it with homozygous recessive parents. Okay, so learning check. We're going to just keep it simple because we'll do more practice in class. Okay, learning check, we want you to do a Punnett square. Predict the offspring of these two beautiful little dogs, or big dogs. Okay, it says assume dad is homozygous, so we'll do dad goes over here. Homozygous means both capitals, okay, because we know that black is dominant. Mom is white, so we know she has to have two recessive alleles. Okay, now to do Punnett square, I'm going to do a B here, capital B here, capital B here, and then I'm going to go ahead and do the next row as well, capital B, and then I'm going to bring the rows, or the columns, and write that down, big B, oh my gosh, I don't know what I'm saying, sorry, little b, and then over here, little b, okay, here, and here, 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 big B, big B, big B, big B, if you're confused about where those came from. Okay, so, oh, and then white is recessive. Alright, so now let's get a little bit trickier. Albinism is a recessive trait, so two alligators with normal phenotypes, so in other words, they are not albinos. Okay, they both have, however, one parent that was an albino. So what are their offspring's chances of being an albino? Okay, this right here, the fact that they both have a parent that was albino, means that each parent had to pass on with them. Let's do big A here is normal, or green, okay, and then little a is going to be our recessive trait for albino. Ooh, I forgot my L, albino. Alright, so because we know both parents were albino, we know that they had to have two little a's. However, since the alligators were normal, okay, then we know that each parent has to have a big A. So we're going to do mom's genotype first, big A, little a, and then dad's genotype over here, big A, little a. So we have two heterozygous parents, and we want to know what are their offspring's chances of being little a, little a. So I'm going to go row first, and then little a. Actually, this should go over here. Okay, so I did my rows first. Now I'm going to do my columns. So I'm going to have a big A here and a big A here. The reason I scratched this one out is because capital letters need to come first. Okay, big A and little a, and look, little a, little a. So only one out of every four of their offspring, one out of every four of their offspring, okay, or a 25% chance that that their offspring will be albino. That's because we have a big A here, big A here, big A here, which means that 3 out of 4 of their offspring, I'm sorry, let me rephrase that. Okay, it means that they have a 75% chance of having a normal green alligator and a 25% chance of having an albino. Okay, and like I said, we'll talk more about this in class. Okay, so now let's take a look at dihybrid. Okay, dihybrid, we're going to set up a bigger Punnett square. Okay, and we're going to look at color and tail length. And I think we're going to have to end here, and we'll just have to finish it in class. So bring these to me, and we will take a look at these in class first thing.